Although there are serious problems in cloning other mammals, there are claims that attempts to clone humans using core transfer are ongoing. Epigenetic and genetic mechanisms are known to be associated with clone failure, but it is still unknown exactly how. Cloning related to human reproduction is not ethical, but the production of cells from cloned embryos can offer many potential benefits. Can human cloning be done safely to achieve this benefit? The mammal that was successfully cloned from the first differentiated animal cell became a sheep named Dolly. Although the previous cloning was successful using embryonic cells, Dolly's unique feature is that it is the first mammal to be cloned from an adult somatic cell. The success of somatic cell nucleus transfer (SCNT), Figure 1, proved that the differentiation of adult cells, in this case derived from the mammalian epithelium, does not require irreversible modification of the genetic material required for animal development. Dolly lived until she was 6 years old and eventually succumbed to a viral-induced lung tumor. 3. Despite these and other successes, SCNT cloning is still inefficient for adult cells. Clones disappear during the early stages of development and during pregnancy. Some clones die prematurely as a result of a number of pathogens. However, some healthy animals were born. But the main distinguishing factors between clones that still do not develop normally and those who won the cloning lottery are unknown. It is a fact that mechanisms involving miscarriages, newborn deaths, and postnatal cloning diseases are less important than the development of core transfer technology. Since it provides stem cells for non-reproductive cloning, research and treatment, Box 1, understanding the causes of cloning problems is highly relevant. A bigger concern is that there are ongoing investigations to some scientists investigating the possibility of human cloning, Box 1. It is vital to address security issues related to this cloning as an urgent issue. All structured and prospective studies are required to identify the mechanisms underlying abnormalities that cause cloning and determining their importance. Non-reproductive cloning involves less ethical dilemmas than reproductive cloning, which may have therapeutic potential in the future. For example, theoretically, diseases associated with damage to organs such as the heart and brain can be treated with cloned embryonic stem cells. Also, using cloned S cells, gene defects can be repaired more easily by homologous fusion. Therefore, the development of these technologies quickly and safely is important in understanding why clones fail. In this article, evidence of cloning failure was considered. Focus is on epigenetic and genetic effects and their consequences on clone development and phenotype. In addition, strategies have been proposed to help understand the mechanisms underlying these four complex processes and to examine the issues in detail. Examining evidence. There are two evidences in the causes of clone failure: species that could not be cloned despite repeated attempts and clone species, all with high clone failure rate. With the development of these techniques over time, it is possible that many species will join from the first category to the second category. However, it is important to consider biological differences in the failure of cloning of some species. Evidence from yet cloned species. Research data from species cloned with SCNT have been tested, but no substantial evidence of mechanisms underlying cloning failure has been obtained. Considering human cloning implications, information from non-human primates is particularly important. Rhesus monkeys (castor) were cloned from embryo preimplantation using nuclei, but their cloning trials using adult cells were not successful. There is now convincing evidence that the meiotic spindle organization and its combination is different in species that have been successfully cloned. Especially during unfertilized egg enucleation, the proteins required for proper meiotic spindle organization, a necessary process before somatic cell nucleus transfer, disappear due to its proximity to chromosome. This data shows that the level of difficulty in cloned rhesus monkeys may be associated with the loss of vital proteins when the cell begins to divide after SCNT. While information from people's close relatives, such as rhesus monkeys, is important, this new study does not prove that similar problems can be encountered in other primates, including humans, when attempting cloning. Also, even if the loss of meiotic spindle proteins during enucleation is a common problem for primates, this problem is likely to be solved by transferring spindle-associated proteins with somatic cell nuclei into the unfertilized egg. At least this study raises the possibility that specific biological differences may be the underlying cause of cloning of primates. Similar causes can be seen under other types of cloning failure, such as dogs. Regardless, cloning is highly dependent on the availability of specific activation technologies. This includes methods of cultivation, activation, microprocessing, and even egg and embryo transfer to physiologically sensitive receptors. To overcome difficulties in the methodology, embryo transfer synchronization and pregnancy care requirements are important in rabbit, pig, and horse cloning studies. The last successful cloning of rats resulted in a method change known to occur in this species, taking into account spontaneous egg activation. Despite the cloning failure in primates so far, he believes that at some stages they have been cloned. Given this possibility, information from cloning errors of the types in which SCNT was successfully applied can be crucial to assess the risks and potential biological effects of the cloning process. Evidence from Clone Species Table 1 provides a brief summary of the major defects found in clone species. Several types of errors have been found, for example, cardiovascular and placental defects, as well as different types of errors have been reported in other creatures, e.g. obesity mice and body wall defects. Reported error severity also varies. In general, defects of mouse, goat and pig clones are less severe than failed sheep and cattle clones. Few studies, however, have characterized complete abnormal phenotypes, and therefore it is not possible to decide their relative severity. Some of the properties presented in Table 1 in ruminants are similar to the large offspring syndrome (LOS) associated with growth and inadequate organ pathologies. This syndrome is not specific to cloning and is associated with embryo manipulations. Some of the features of the LOS phenotype, especially overgrowing body wall and musculoskeletal system defects, increase in children produced by in vitro fertilization, similar to Beckwith-Wiedemann syndrome in humans. It is clear that some of the defects found in these failed clones are likely due to embryo manipulation rather than cloning. Finding cloning-specific causes requires careful experimental planning and clever use of controls. With few exceptions, reports of abnormality in cloning focus largely on surviving fetuses, because fresh tissue required for reliable and histological evaluations is not available in clones that fail early. Nevertheless, several studies examining abnormalities that occur early in development have identified significant placental pathology abnormalities. In particular, some unsuccessful ruminant clones have insufficient placental development, and placentomegaly resulting from expansion of the Spongioblast layer has been reported in clone mice. The more extensive characterization of these placental malformations undoubtedly allows us to understand high fetal losses and may indicate that other phenotypes identified, particularly cardiovascular and pulmonary lesions, reflect secondary phenomena rather than directly caused by genetically induced defects. Genetic and epigenetic effects of cloning. Now, there are a few ideas about some types of pathology that occur in cloned mammals, but what is the responsibility of epigenetic and genetic effects in the development of these pathologies? Although there are signs of chromosomal abnormalities, the evidence of genetic effects in clone failure is limited. However, the fact that clones with an abnormal phenotype do not transmit abnormality to offspring produced by natural mating is direct evidence that inappropriate gene expression is the result of errors in epigenetic mechanisms. These mechanisms allow molds that allow DNA or chromatin structure to continue to be conserved in hereditary but reversible modifications in sister cells that arise from meiotic cell division. These include post-translational protein modification and DNA methylation through mechanisms such as acetylation and methylation. See source article for further reading. Interpreting the facts. Significant contradictions emerged in the interpretation of these observations. These will polarize either against those who advocate human reproductive cloning efforts or those who find hazards in nature unacceptable. Another point of view is that non-human therapeutic cloning is acceptable, but reproductive cloning is unacceptable. We are against any attempt to human reproductive cloning, and we emphasize that the ethical and moral issues surrounding such a study should immediately attract the attention of the scientific community. Discussion advocates of human cloning have suggested that cloned animals appear healthy, and that the reported abnormalities are similar when compared to normal reproduction, even at a higher frequency. It has been found that the success rates reported in some studies are much higher than others, and many studies on adult surviving clones show no anatomical or physiological abnormalities that can be detected by routine diagnostic methods. Also, although there is abundant evidence for abnormal reprogramming and altered gene expression profiles, the identification of regular expression profiles for several genes in some cloned embryos can be discussed. For example, metabolic enzymes lactate dehydrogenase, citrate synthesis and phosphofructokinase, October 4, GP130, TEC3 and RNA polymerase and beta-actin GFP in swine. In addition, genes responsible for some epigenetic modifications, such as reorganization of telomere length and X inactivation occurring after the zygote stage, have been efficiently reprogrammed in some cloned animals. Clone animals that are apparently healthy and reach adulthood can be produced, but on the contrary, clones are often characterized by the extreme errors described above. Human cloning promoters argue that only the safety test can be allowed to develop depending on the screening of embryos in embryo selection. For example, cells can be removed at pre- and post-implantation stages and evaluated for imprinting errors and gene expression. However, given that all of the potential negative epigenetic effects should be known, reliable preimplantation selection is currently not possible. In contrast, preimplantation diagnostic strategies scan genetic effects known. Moreover, given the developing embryos shown to be mosaic in nature, the analysis of single cells from embryos will be wrong. It is important to emphasize the difference between the embryo selection used in genetic diagnosis before implantation and the embryo selection used to ensure the normality of the clone. Existing techniques cannot guarantee the latter, therefore, the existing techniques currently do not have the reliability capacity to estimate the development performance of embryos, except for pre-implantation embryo analysis for specific genetic defects. Result experience with other mammals shows that any attempt to clone humans is now naturally insecure. Scientists should not tolerate human reproduction cloning for these reasons only, without taking into account equally important moral and ethical values. Still, cloning has the potential to contribute to animal productivity and health improvement. There is also the possibility that non-reproductive human cloning strategies may provide future treatments for incurable diseases. However, this goal will be achieved by knowing more about the basic molecular mechanisms involved in core reprogramming. A better understanding of these mechanisms will provide insights into the importance of epigenetic events involved in the development and provide new approaches to improving the efficiency and success of core transfer procedures. Concepts in Translation Parts Somatic Cell Core Transfer (SCNT). In this process, the nucleus of an adult cell is transferred to an enucted cell, the reprogrammed oocyte is activated, which initiates further development. Epigenetics. Literally, outside of classical genetics, this term describes hereditary changes in gene expression, which is not caused by a change in the DNA sequence. Go phase. The resting phase of the cell cycle. The enucleatio. Core removal. Blastocyst. A preimplantation embryo containing a fluid-filled cavity, blastocele, a focal cell cluster, inner cell mass from which the embryo will develop, and peripheral trophoblast cells. Big puppy syndrome. A syndrome characterized by fetal length and various organ pathologies. Overgrowth. Increase in body size and organ size compared to normal controls. Inner cell mass (ICM). It includes indifferentiated cells, a small group within the blastocyte. Morula. A preimplantation embryo consisting of a solid cell cluster. Placentamegaly. The growth of the placenta beyond its normal size. Spongioblast. The outermost structural layer of the placenta.